Hello everybody, this is D. Rajashekar, lecturer in botany, Srimati Ganganeni Kalyani Government Degree College, Vinikonda, Guntur district. In this video lecture, I would like to give structural details of bacteria. If you observe bacterial cell structure, you can come to know the following facts. Generally, bacteria can be seen under compound microscope, but the structural details can be seen only under electron microscope. Bacteria is a plural form, and the singular form of bacteria is bacterium. Bacteria is a unicellular prokaryotic microorganism. That means it consists of only single cell. That is the organism and that is the cell only. And prokaryotic microorganism. Prokaryotic means in the bacterial cell we can see the nucleus without nuclear membrane. So it is not intact one. Such nucleus is called nucleoid or genophore and incipient nucleus. Hence it is called prokaryotic microorganism. Pro means primary, karyo means nucleus. Since it reproduces by binary fission, it is put under the class Schizomycetes and the order Monera. Bacteria was first discovered by a Dutch scientist by name Antoine van Leeuwenhoek. And when he first observed this organism, as it was showing movements like other animals, he called them as smallest animals or animalcules. It was Ehrenberg first named them as bacteria. Bacteria grow everywhere if they find room temperature and neutral pH. They can grow very easily. That is why they can be seen in every aspect like soil, water, hair follicles, organisms, outside the organisms, inside the organisms, everywhere. That is called omnipresence. So bacteria is a omnipresent microorganism. The study of bacteria is called bacteriology. Bacteria cell ranges just a few microns. All the bacteria cells measure 1 to 10 microns length and 0.0 to 2 microns diameter. The smallest bacteria we know is Mycoplasma genitalium. It ranges just 200 to 300 nanometers. And the bigger bacteria is Thiomargarita namibiensis. It is a gram-negative and coccal type of bacteria. It ranges 100 to 300 micrometers, whereas Epulophisium fischelsoni is a biggest bacillus type of bacteria. In this video lecture, for better understanding the cell structure of bacteria will be studied under two heads. One is the cell organelles present outside the cell wall and the cell organelles present inside the cell wall. Generally, the organelles which are seen outside the cell wall are capsule, glycocalyx, flagella and pili. Whereas the cell membrane, mesosomes, cytoplasm, nucleoid, plasmid, gas vacuoles, reserve food materials come under the organelles inside the cell wall. Bacteria cells appear in 4 different shapes such as coccal forms, bacillus form, vibrio form and spirillum type. As you see in this slide, different bacteria shapes. The spear shaped or round shaped bacteria are called coccal forms. If there is only one round shaped bacterial cell, that is called monococcus type. And if you see two round formed structures present one beside another, that is called diplococci. If you see more than 2 round shaped cells present one after one like a chain, that is called streptococcus. If such round shaped forms are present like a grape bunch, that is called staphylococci. Similarly, if there are 8 round shaped bacteria present like a cube, such a shape is called sarcina. Similarly, the another shape of bacteria is rod shaped bacteria, that is also called bacillus type. If we see only one rod shaped bacteria, that is called monobacillus. If we see 2 rod shaped bacteria present one after one, that is called diplobacilli. If we see more than 2 rod shaped bacteria present like a chain, that is called streptobacilli. Similarly, there is another form that is called comma shaped, that is also called vibrio. And the fourth one is spirillum shape. Here we can see, spiral shaped bacteria. Example, spirochetes. Spirochetes show more than one spiral. And there is another bacteria which is called pleomorphic bacteria, which can show different types of shapes. That is called pleomorphism, and those bacteria are called pleomorphic bacteria. Now we focus on the cell wall. Basing upon the cell wall constituents and a staining procedure, that is called Gram staining procedure, which was actually developed by Christian Hans Gram. By his experiment, basing upon the cell wall constituents, bacteria are broadly classified into gram positive and gram negative bacteria. Generally, gram positive cell wall is thicker than the gram negative bacteria. In gram positive bacteria, we can see mucopeptides, peptidoglycans, and teichoic acid. Peptidoglycans are 95% of all other constituents of cell wall in gram positive. These peptidoglycans show 2 types of subunits, sugars and amino acids. The sugars are of 2 types, NAM and NAG. NAM means N-acetyl muramic acid and NAG means N-acetyl glucosamine. Teichoic acid also present. Generally, these 2 sugar molecules are present one after one and form like a long chains, and they are connected by the peptide chains. And we can also see teichoic acid molecules. In some other bacteria, they are popularly known as acid fast bacteria. They also show some other chemical composition that is called mycolic acid. They are Mycobacterium tuberculosis. As they are stained with acid fast stains, they are called acid fast bacteria. Generally, gram positive bacteria are non-pathogenic, but there are some gram positive bacteria which are with more teichoic acids and mycolic acids. They show pathogenicity. Generally, gram positive bacteria secrete exotoxins. Because of this, they can get the pathogenic nature. When you come to the gram negative bacteria, they show 5 to 10% of peptidoglycans. Teichoic acid is completely absent. Along with the regular and thin peptidoglycan, we can see outer layer, which is generally made up of lipopolysaccharides, lipoproteins and phospholipids. Because of this outer layer, gram negative are highly pathogenic than that of the gram positive. And these gram negative bacteria also secrete exotoxins and endotoxins. So as we discussed that Christian Hans Gram developed a staining technique for bacteria. After application of that staining technique, because of the difference in the cell wall constituents, bacteria are classified into gram positive and gram negative. So in the gram staining technique, if you see the bacteria in purple violet color after the staining process, they must be called gram positive. If the bacteria appear red in color after the staining procedure, then they must be called gram negative bacteria. In some bacteria, outside the cell wall, there is an outer layer made up of polysaccharides. It is an extra protective layer. Generally, cell wall is an extra protective layer. Along with production, it also gives a shape and size to the bacteria. Whereas these outer layers are providing extra protection to the bacterial cell wall. If the outer layer is a thin and a loose layer, then it must be called as slime layer. If this outer layer is somewhat thicker, then it must be called as capsule. Generally, pathogenic bacteria show capsule. The slime layer or capsule, as we discussed, they provide extra protection for the bacteria. They protect the bacteria from desiccation. That is why bacteria can withstand even at high temperatures. And the slime layer-like structure helps the bacteria to attach a substratum. And this capsule, also helps the bacteria to endure all type of unfavorable conditions. So as I told you in the beginning slides, now we are discussing the outer cell organelles present outside the cell wall. Of them, flagella are very, very important. Flagella are locomotory organelles. They are very thin, thread-like appendages. Actually, they extend outward from cell wall and plasma membrane. They are very slender structures, ranges 15 to 20 micrometers only. Generally, these flagella of bacteria are made up of a special protein called flagellin. They show three parts, like basal granule present embedded in the membrane, shaft, hook, and the shaft. Shaft is like a whip-like structure present outside the cell wall. Generally, bacillus forms and spirillum type of bacteria show flagella than that of the other type of bacteria. Basing upon the arrangement of flagella, bacteria are again classified into 5 different types. There are bacteria which do not show even a single flagellum. Such bacteria are called atrichous bacteria. Example, lactobacillus bacteria. Then monotrichous. Some bacteria show single polar flagellum that can be seen only at one end of the bacteria. That is called monotrichous bacteria. Example, Vibrio cholera bacteria, Pseudomonas aeruginosa, etc. As you see in the picture, it shows only single flagella at one pole. Then Lophotrichous bacteria. Here you can see cluster of flagella at one end, as you see in the slide, especially the second structure. Lophotrichous type of bacteria, flagella, can be seen in Spirillum type of bacteria and also Pseudomonas fluorescens. Then Amphitrichous flagellum. Here you can see single flagellum at both ends. Example, Aquaspirillum. And similarly, Peritrichous bacteria. Here flagella are seen all over the surface of the bacteria. That is called Peritrichous flagellum. Example, E. coli bacterium and also Salmonella typhi. In eukaryotic organisms, the flagella show 9+2 arrangement of microtubules. The 9+2 arrangement of microtubules cannot be seen in the bacteria flagella. And pili or fimbriae. There is small difference between the fimbriae and pili. Fimbriae are short appendages, they are tube-like structures, whereas pili are somewhat long than that of the fimbriae. They are also tube-like structures. Generally, they are seen in gram-negative bacteria. Normal pili or fimbriae are mainly meant for the attachment of the substratum only, whereas the pili, which help the bacteria during conjugation, that is transfer of genetic material from one bacterial cell to another bacterial cell through conjugation tube. For conducting conjugation, the special pili are developed. They attach the other bacteria so that the gene transfer can be easily happen. Such pili which are helping in the sexual reproduction like process are called sex pili. Pili or fimbriae are generally made up of a special protein called pilin. So far we have discussed the structures outside the cell wall. Now we will see the structures inside the cell wall. As I told in the beginning slides, inside the bacterial cell you can see, after the cell wall you can see plasma membrane, a thin wall structure. It is made up of lipids and proteins which show the arrangement as Singer and Nicholson proposed theory, that was a fluid mosaic concept. This plasma membrane is semi-permeable membrane present below the cell wall. There is a space actually between the cell wall and plasma membrane that is called periplasmic space. This plasma membrane folds inwardly in the bacterial cell. These infoldings of plasma membrane is called mesosomes. Meso means middle, zoma means body. Generally, these plasma membrane infoldings are seen in the middle of the cell wall, middle of the bacterial cell. Hence, they are called mesosomes. Mesosomes generally they show respiratory enzymes, so that they may be involving in the respiration activity. They also involve in the equal distribution of genetic material between the two daughter cells during binary fission. And then the cytoplasm, below the plasma membrane, the inner liquid part of the bacterial cell is called cytoplasm, which consists of minerals, reserve food materials, nucleoid, plasmid, polysomes, reserve food material, carboxysomes, magnetosomes and gas vacuoles. The liquid part of the bacterial cell structure is broadly called cytoplasm. Then the major part is nucleoid. In the beginning itself I told you. So when we see the bacterial cell structure, you can see a circular molecule is a double helical structure, that is a DNA. It is not enclosed by any nuclear membrane, so that it is very incipient. It shows only DNA molecule, hence it is called nucleoid or genopore. It consists of all genes which are essential for the metabolic activities of the bacterial cell. Along with this regular chromosome or nucleoid or genophore, we can see an extra chromosome in the bacterial cell. That is called plasmid. This plasmid is an extra chromosome. It is also a circular double helical structure. It carries special genes like antibiotic resistant genes. Similarly, NIF genes and some special genes are seen in the plasmid. Moreover, plasmid is an independent genetic material which never undergoes the control of nucleoid. And one more advantage of plasmid is when we join a foreign DNA or foreign genomic material, it takes it for granted, and along with its replication, it also replicates the added foreign genetic material. That is why it is widely used in the genetic engineering techniques. Then polysomes. If you see the bacterial cell structure, in the cytoplasm, you can see small granule-like structures which are closely adjoined together called polysomes. Individually they are called ribosomes. We can see small subunit and larger subunits of ribosomes, but generally they are present in groups called polysomes. Ribosomes are mainly meant for protein synthesis. Here in prokaryotic organisms like bacteria, the ribosomes are of 70s type. The small subunit is always 30s, whereas the large subunit is 50s. Then the reserve food material. In the bacterial cell, reserve food material is stored in the form of PHB granules or diluting granules and glycogen. PHB means polyhydroxybutyrate granules. In some photosynthetic bacteria like chlorobium, chromaceum, green purple sulfur bacteria, we can see small green color structures called carboxysomes. They are like chloroplastids. They are called precursors of the chloroplastids, what we see in the higher plants. In bacteria, we can see carboxygens instead of chloroplastids, precursors of chloroplastids. So that these special bacteria which have carboxygens can perform photosynthesis. Hence, they are called photosynthetic bacteria, utilizing light of the sun and consuming CO2 from the environment. With the help of these carbogens, they can prepare their own food materials. In some bacteria, we can see special structure. They are called magnetosomes. They have magnetic properties. So the bacteria which have these magnetosomes are called magnetotactic bacteria. So with the help of these magnetosomes, they orient in north and south directions, maybe for better life. In some bacteria like holobacterium or aquatic bacteria, we can see small vacuole-like structures filled with gas. These are called gas vacuoles. These vacuoles are, as they are with gas, they provide bacteria buoyancy, so that the density of the bacterial cell is decreased, so that they can easily float on the water surface. So these are the different structures or cell organelles we can see inside the cell wall of bacteria. So before closing this video, I would like to recall the few important aspects of bacterial cell. Bacteria is a plural term of bacterium. It was discovered by Anton von Leeuwenhoek and named by Ehrenberg. It is a prokaryotic single cell microorganism. It is omnipresent. It ranges few microns only. The cell structure, basing upon the cell size, bacteria are classified into 4 different shapes. They are cocal shaped bacteria, bacillus shaped bacteria, comma shaped vibrio bacteria, and spiral shaped spirillum. Again, they are classified into different subtypes. For better comprehension, the bacterial cell is classified into cell organelles present outside the cell wall and cell organelles present inside the cell wall. Thank you for your patient listening. We will meet in another video lecture. Till then, bye. Thank you. Thank you one and all.